I would like to show you the program shortly. We will having like opening ceremony for like 5 minutes, and then we will start with Professor Dr. Alpan Bek, and then Professor Hassan Yilmaz, then Professor Rashid Turan, then Professor Buland Ortaç. Between each presentation, we will have break, and these breaks are meant for socializing and like talking with professors and having like casual talks. After each presentation as well, we will have like a question answer session. At the same time, we can have the chance to discuss our questions with the professor immediately. At the end of the presentation, we will have a surprise lottery, and we can provide attendance certificate for those who would like to. So stay till the end, and we will send you by email your attendance certificate. I would like to give the words now to Jemre. Jemre is a third-year student at Physics Department in METU, and she is the responsible for the student chapter now. I would love to give her the word to welcome us. Jemre.
Hello, everyone. Thank you for joining us in this very special event. We have been preparing it for weeks, and we are very excited about it. We are also an SPIE student chapter in METU, and we are also preparing to be the Optics Photonics Society in our school. And OSA is Optical Society, and SPIE is Society of Photo-Optical Instrumentation Engineering. So they're both prestigious and rooted societies, and we are very proud to be their student chapters in METU. And we usually prepare seminars and panels with our professors, and we are happy to have you as member. Thank you for coming today again. Thank you, Tarık. And I can introduce you Professor Alpan Bek as the first speaker. Welcome, sir.
Hello, welcome. Welcome to all. Thank you for introduction. Maybe I should make a small introduction of myself, or do you have anything to introduce, Cemre?
Actually, we had, but I'm sorry, Tarık was going to do that.
Okay, you can go ahead, Tarık.
Yeah, I have to, but like I have a problem with my camera now. I don't know why it does not show up. I'm really sorry about that.
All right.
Okay, no problem. We have our Professor Alpan Bek. He is associate professor in the physics department at our Middle East Technical University, and he is the leading member of the nano optics laboratory at the department, and he is a faculty member of the master's and PhD program of MNT, Micro and Nanotechnology. At the same time, he had like his education was from Bilkent University, his bachelor and master's degree, and he went to Switzerland in the university in the School of Polytechnique Federal of Lausanne and Switzerland. He did his PhD there. After that, he like went to several places in Max Planck Institute and Ludwig Maximilians University of Munich, and other research in other several areas. His talk about behavior of Neuro-2A cells of femtosecond laser micromachined silicon substrates, and he will talk about it in his presentation. So welcome, Professor.
Thank you, Tarık. So I'm also the supervisor of the SPIE chapter of METU. As we come together here as an event which is organized by the, I think, two communities, two student chapters of Bilkent and METU, right? So there might be those who have not been introduced to the chapters before. If there are those audience who are not member of the chapters, I strongly recommend you to become a member and be active, as this is a career-oriented initiative derived from students, and it has all the point of view from the student eyes and not from a top-down instructed organization. So it might be very helpful for you to draw your own career line in the future, in the near future, and please take part and be involved, and you will benefit from these chapters to its max. So as a supervisor, that's all I want to tell. As a speaker, today I have prepared you a talk which is based on, which is actually an application of photonics, not a hardcore photonics development, but it's an application of our photon tools that we have available at hand in our laboratories, how these photonic tools can be applied in benefit of human health and therefore society. So this talk is going to be a kind of introduction to that sort of research. So it's partially biophotonics, partially biology, and also partially completely photonics. I'll start by sharing my.
Let me stop my share, Professor, and let me add something small. This presentation will be recorded, but we will only record the speaker and the slides. So all of the students can feel free to open their cameras and interact with the professor immediately. That would be really good.
Yeah, if you have questions, please feel free to interrupt and ask it at the point. Don't wait until the end. Ask it as the topic is hot. Okay. All right, so I can start sharing, right, Tarek, right now?
Yes, you can.
Okay. Let me see. Does it look nice?
Yes, it looks very nice.
Okay, I'll hide the video panel. So, all right. Now, as you see this talk, although I'm the speaker today, but it's work in which many researchers were involved. You see the names here. Sara Mingu was is my former master's student. She graduated. She's right now doing her PhD in a center in Mainz, in Germany. Ihor Pavlov is a faculty member of physics department. Çağdaş Devrimson is a faculty member in biological sciences department in METU. And we are associated with both the physics department and micro and nanotechnology graduate program, Center for Solar Energy Research and Applications. And today I'll be talking to you about the interesting behavior of Neuro-2A cells on femtosecond laser micromachined silicon substrates. So this is a photonic community. I reckon that the second half of the title is more relevant to most of you, femtosecond laser micromachined silicon substrates. But that's why I would like to give more information about the Neuro-2A cells and their behavior, why they're important to you, to show you how we can help by using our photon tools. In photon tools, what I mean by photon tools is this application of femtosecond lasers for micro machining the surface structure of silicon and several other materials. To guide neuron cells or stem cells, which can be differentiated into neuron cells, to do their work. Okay, so in that respect, it can also give you an idea what sorts of applications do exist for strong pulsed ultra short lasers. Okay, now it's a question why we should study cells on different structures or topography, surface structures and topographies. Well, the reasons are multitude. The topography or the surface structure, this is what I mean by topography, the surface structure on which the cells are grown do affect the adhesion properties of cells, their multiplication, proliferation, biological term, their shape, three-dimensional shape, how much the stem cells can differentiate into certain tissue cells, differentiation, how quickly they move, where they move, and in which direction and in which sorts of motion, exploratory motion, they do. So these cells obviously are live material entities, and they do explore their environment, and surface topography can interfere or affect, let's say, with their migratory or exploratory behavior. You can see many different publications which are, how to say, these are literature review articles in which you can read more about these properties of surface topography in cell behavior. Okay. Now, let's take a look at several applications. Surface topography can be used to control cells on substrate in several ways. For example, cells can be patterned to occupy certain locations more densely than the rest. This could be used for example, construction of neural networks. And they can be used to, the surface topography can be used to control on how far the cells, stem cells, can differentiate into neuron cells. It can be used for the proper alignment of the cells. Sometimes, many of the times, neuron cells are elongated cells. They are not spherical, but they're elongated. They form axons. I'm sure all of you know about these axons. And also there are philopodia. These are little feet with which the cells move around. And the alignment of these philopodia and their axonal extremities can be directed by surface topography. They can also be used to improve certain properties. For example, we can make use of the topography to engineer smarter interfaces with the cells. Their contact areas with the surface can be modified. You see surface structuring at the micro and nano scale does affect the cell hydrophilicity and hydrophobicity. Okay, many of the super hydrophobic surfaces do have highly textured, nano-textured surface shapes and topographies, and in a similar sense, cell adhesion can be modified. It's not exactly hydrophilicity, hydrophobicity, which is involved here, but in an analogous sense, cell adhesion and its willingness to adhere, to stick to the surface, can be modified by using different densities of nanotexturing at the nanoscale and microscale. This can help us to construct smarter interfaces. The surface topography can allow also for fabrication of chips, silicon-based biological chips, assays in which alignment and growth of these cells can be used as a detection signal for identifying certain cell lines in between other cells. Okay, so cell tissues grow in natural organisms side by side. They are very closely intervened. They are not isolated as in the lab petri dish. So if you extract tissue cells out of an organism, you may want to purify or let's say separate them. And this separation can be, for example, made based on their size, their surface, their shape, etc. And also they can be separated by means of their reaction to the surface, to a certain surface. And with surface, what I mean is certain surface texture. Some of the tissue cells like to align more in the direction of the surface topography. Some of them don't. And this can as well be used to separate different cells extracted from an organism. Now, with why we use a Neuro-2A cell line? Well, what is a Neuro-2A cell line? It's a mouse neural crest-derived cell line, and it's being used very extensively for neuronal differentiation, axonal growth, and signaling pathways. One important thing about this cell line is that they don't require extensive durations in order to experiment. They allow for studying many different effects on them within only a few days, which makes the cycle of a scientific study, life cycle of a scientific study to be as short as a couple of days, maybe 3 days or so. Otherwise, normally the biological cell lines typically require extensive times for their growth, for their conditioning, etc. But this Neuro-2A cell line is rather quick, rather fast. So one feedback cycle doesn't take too long time as compared to other biological cell lines. So it's a stem cell line which has potential to differentiate into various different kinds of neuron cell lines. Okay? It's a specific stem cell line for neuronal cells. They're widely used on different materials. The literature is rich. Their aligned neurite are these extremities which start to grow out of the central body of the cell. Okay, their alignment is important for regenerative medicine applications. Like if you have a neural damage, you can think of implanting the cell lines to let them differentiate into the missing cells in your neural system. And they are commercial. You can purchase them. They're highly available and they're easy to manipulate in the lab. So then, as I said, we will study their behavior on silicon, but many different materials can be textured by using lasers. Why do we use silicon? Well, it has many superior qualities. It has a native oxide layer on the surface, which makes them rather inert and protected against external effects. They have many different applications, electronic applications and photonic applications, which can be used to interface for biological systems with the electronic systems. We haven't done that, but it's possible. You can construct electrodes on them. You can bias the cells, give some pulses to cells using the substrate, etc. And their silicon's modification or topographical behavior has been extensively studied. It's very well documented material, and it's not toxic to cells. So it's inert, it's not harmful to cells, and it can be shaped into many different topographies. Okay? So let's see how we go ahead. So we start with.
Before going further, I just wondering about something. Can I ask? Because you
Sure, of course.
stopped. I'm wondering about, you said that because of the topology, we can differentiate these cells. Can we differentiate, for example, if we can use further this technology to differentiate the brain cells from each other? I mean,
different neuronal cells from each other.
All brain cells, for example, but differences between them too, if they have, we can.
So, brain, thanks for the question. Brain tissue comprises of, I don't know, I'm a physicist, but I think 20 different tissues. And many of them are kinds of neurons, but there are also some supportive cells, ganglions, etc. And if you would like to differentiate between these cells, to start with, let's say separation of these different kinds of cells, there are, first of all, simpler things that you should look at than trying to use surface texture to differentiate them. The first thing you can do is to take a look at the size. Okay, you can use many different, much easier under the microscope techniques to separate them. But then there are certain lines which don't show a clue about what sort of cell lines they are just under the microscope. In order to figure out about the types of these cells and separate them from each other, yes, you can, for example, start with a certain topography of silicon at nano or micro scale, and you can check the tendency of different cells to adhere to the surface. And this tendency for adhesion to the surface can give you a clue, and it can be used to actually separate certain cells from the rest. Okay, but there are many different things one can do as the growth, or let's say as the time that these cells spend on the substrate increases. For example, their speed, their viability, how well they survive, etc., can be used to separate these different types of cells. Yes.
Also, I'm wondering about, I think that you just only consider the topological things, but can we do some other experiment on the chemical or electrical transferring between the neuron, and if we use the metallic layer with the laser, we can morphologically change the metallic layer, and can we see the electrical transference between these two things, I mean, layer between the neuron to do metal? I mean, just asking the questions because of the, if we can, adhere to brain cell, and then because we know that the brain cell use the electrical things, electrical signal, yeah. And then because of this, maybe electrical signal can pass through the metal layer, then maybe we can use the for example.
Yeah, neurons do have this particular behavior when they are in a network. If they, I mean, I'm talking about well-established, differentiated, and well-grown neurons. When they are, the cells that I talk about are not neurons, they are stem cells which can become neurons. Okay. But for the neurons, well-advanced neurons, they survive only if they receive pulsed signals, electrical voltages. if they don't receive electrical voltage pulses, they decide to die. They decide to commit suicide, because the purpose of a neuron is to transmit information. And information transfer is in the sense of voltage pulses. Okay. If the neuron cell doesn't receive any voltage pulses anywhere and doesn't conduct this pulse to anywhere, it tells it's interesting suicide process starts, and the cell decides to commit a suicide, which is called apoptosis. So now, in order to do experiments on the voltage pulse conduction and use this to separate the cell behavior, the cells have to be well grown into fully functional neurons. Okay, and in the earlier times, as in the stem cells, these voltage pulses, they don't need the voltage pulses to survive. They just survive as they are because they are kind of dormant. They wait for their time to become neurons, okay, when they are needed. So therefore, electrical signals and these voltage pulses, I don't think it's going to be too much beneficial in the early stages of these cells. But yes, it can be used to let certain cells behave in a certain direction as the cells become fully mature into neurons. Okay. So, for example, in construction of a neuronal network, this pulsation of voltage pulses could be a key to keep the cells alive and fully functional. Okay? So in that sense, yes, you may want to have a metallic interface between the cell and cell substrate and the cell as an use as an electrode. And typically, there are many inert, let's say, biocompatible metals which can be used as electrodes, such as titanium, etc. Okay. So let me go ahead with the methodology. You're welcome. We start with substrate preparation. We structure the substrate surfaces. We first, of course, do a cleaning, and we characterize the surface. We sterilize the surface for cell growth. Then we implant the cell culture onto these structured surfaces. And then as the cells are grown into certain phase, the phase durations are given here, indicated here, 3 hours, 24 hours, 48 hours in our study. And after 3 hours, we prepare the cells, for example, by labeling them with fluorescent dyes, which enables us to image them under fluorescence microscope. And then we transfer these cells onto we transfer some genetic material into these cells, if required, by so-called transfection, to enable them to express certain proteins. And these proteins can behave like fluorescent markers themselves, such as the green fluorescent protein, yellow fluorescent protein, etc. Okay, so those are typical cycle of preparation. Then, of course, once we are ready to go, we start our timer. We wait for a certain duration of time for the cells to grow, to let them freely grow on these surfaces. And after these certain durations, we start visual inspection of our surfaces. Of course, for live cells, which are marked by fluorescent dyes, we use fluorescence microscopy to study the motility and their exploration behavior, their spreading behavior, how they spread, how quickly they move, how much they explore their environment, etc., on live cells. One other kind of research you can do on these is you can kill the cells instantly, fix them, and then you have a kind of snapshot of that moment. Okay, the cells don't move anywhere, but they are fixed. You can use, for example, more harsh sort of characterization techniques, for example, scanning electron microscopy, to inspect that snapshot in the mold, in the evolution of the cells. Why do we need to fix the cells? Because electron microscopes don't like live cells. You cannot support live conditions in an SEM chamber, electron microscope chamber. It's under vacuum, and the humidity is removed from the cells. The cells have to be therefore sacrificed, so to speak, and fixed in order to inspect under the SEM. With that, we get statistical information about the number of cells, their shape, their alignment. And you have to do this sacrifice and fixing of the cells at various different durations. And each of the sample is going to correspond to a different lifetime of the cell. And then you can study the statistics in let's say, ex vivo conditions. Okay, but it will still give you an idea of what the cells do in their lifetime. So let me go ahead with the laser structuring. Laser structuring is performed in a self-organized and very rapid and direct manner. We do have an infrared laser, pulsed infrared laser at the femtosecond. And after certain conditioning about the beam, based on its direction, polarization, etc., the beam is directed into a galvo mirror scanner. This scanner has two mirrors which are mounted on galvo scanners. Galvo scanners are rapidly moving tip and tilt stages on which relatively low mass tiny mirrors are loaded. And these mirrors are mounted in a way that one of them tips and tilts in one direction, the other one tips and tilts in the other direction. Okay? So by combination of this tip and tilt of the two mirrors, a two-dimensional array can be scanned. And then this image shows the scanned behavior here in C and here in C. So you see the pulse starts from here, goes there. This is our representative focal spot size. You see that within a focal spot, these multitude of parallel lines do start to form on the surface in a completely self-organized fashion. So our spot size is much larger than the actual features we write onto the surface. Why such tiny features are written on the...
Now it looks good.
Now looks good. Okay, let's hope that it will keep like this. So, okay, by raster scanning the beam, we are able to structure extensively large areas on the surface. You see a typical SEM image of such a surface, structured surface, and this is an optical image showing nice coloration due to photonic effect of these parallel structures also here in this image. Okay, so what does the laser look like? Well, it has many components. I will not go through them. This is not the laser that we have used to structure the surfaces. This is an example of such a laser. And the laser that we have used to structure the surface is 1030 nanometers wavelength in infrared. It has several hundred femtosecond pulse duration, but it's a fiber laser. And a fiber laser, this is another kind of fiber laser. This is for 155, it's nanosecond, but a typical fiber laser consists of a gain fiber, and it's all fiber elements. So the light travels within the fiber during all the gain and structuring and shaping and stretching and amplification part until the end of the collimator. The beam is taken out into free space and is sent to the scanner stage. Okay, so this is what a typical pulsed fiber laser looks like. For details, you can check this paper. Now, this is a video of live video of such a surface texturing scheme. You see here a piece of silicon. These are previously structured locations, and in this spot you see a live structuring of a certain region, square region. You see how rapidly and quickly a large, relatively large area can be structured, textured using such a laser. You also see a slight fume coming out. So there is definitely some material removal from the surface, okay? So our laser is, as I said, a 1030 nanometer wavelength, 1 megahertz repetition rate, 370 femtosecond, and it can provide up to a watt. There is a motorized stage and a galvo head for scanning a large, or let's say structuring a large area. With this method, we can fabricate the texture directly on the surface. The surface does not require any further texturing or processing. It just requires maybe some cleaning and conditioning for growth of the cells, like sterilization, etc. But otherwise, it doesn't require any further fabrication steps. This technique is called laser-induced periodic surface structuring, LIPS in other words. It's very versatile, it's very fast. You can choose to structure a square or a rectangle or whatever arbitrary shaped region on the surface. And speed of processing can be adjusted to give you the most beneficial structure that you like. For example, if you scan very quickly, rapidly, you end up with nice parallel lines with several hundred nanometer periodicity. But if you scan too slow, you can structure the surface further into larger, much larger structures, into microscopic structures, which are called microcolumns, etc. We will see. And this can be applied both in atmosphere or in a desired liquid if you want. So here are several different topographies which can be achieved by using this technique. In the first two A and B panels, there are LIPS regime in which you see a representative focal spot size marked by this circle. And within the focal spot, you see at least 6 or 7 lines or 6 or 7 periods. So that means the structures are not written one by one, as if a simple, smaller spot carves these lines out. It's not like that. Within one spot, these parallel lines form automatically, completely by themselves, okay, in a self-organized fashion. When you move the spot, 7 or 6 lines do start forming continuously at the same time. Then you move back and back and back, you construct a large area surface. Now, if you slow down a bit, this structure can be forced into much larger and less organized, crowded structures. Here you see a micro column. So the much more power is incident on this surface than this surface. And this bar shows 2 microns. So our features are of the order of several microns, as you see here, from micron to several microns. And the regimes which we use for achieving lips and microcolumns are shown here. And what you can combine these two as well. Here is an example. At the outside, you see a lower period region, and in the center, you see a micro column region in which the feature size are much larger. But outside, there is several hundred nanometer periodicity. You can as well use this laser in a classical fashion in which you really ablate quite a lot of material and scratch the surface in the classical sense. If you slow down even further, okay, you can open a trench line, deep trench line at the micro scale. This is sometimes lips is confused with the classical laser scribing. So these two images show the big difference, okay? Lips technique produces much finer and small period surface and much more organized surface topography. Classical laser scribing generates a lot of debris. It makes a huge crater, like a micro scale crater, and you cannot produce them as high in a small period as you can with the lips, okay? Now let's take a look at the cell growth results. These are 3 to 6 hours of incubation results. What we see here are, again, SEM images. As I told you before, the cells have been here sacrificed and they are fixed, so they are not live. But nevertheless, they give you a good idea of what different forms of cells can be found on these different surface topographies. For example, this is a close-up image of a cell which was grown adhering to the lips surface. And this is an example of a couple of cells probably adhering to a microcolumn surface. You can see some axonal, or let's say neurite growth, which focal adhesion points, which these cells grow to use to attach to the surface. Where, as you see here, the cells look more uniform, more round. They don't need to grow extremities in order to adhere. When you have a structure which is mixed between lips and microcolumns, you see some regions in which the cells prefer to adhere to microcolumns rather than the lips regions. So it turns out that cells prefer the microcolumns to adhere to microcolumn structures more than they do prefer to adhere to the lips surface regions. You can do the test against flat, completely untouched surface, flat silicon versus microcolumn, and you see that yes, cells do prefer mostly the microcolumns, but also some of them prefer to stick to the flat surfaces. In comparison, when you look at the cell adhering to a flat silicon surface, you see they are less round, they are more grown into these exploratory and growth growth cone kind of extremities, as if they have been more mature during their growth. So now let's take a look at the instruments for characterization. Well, we use an inverted optical microscope. It's a life science edition. We use a light source and we use a camera. Light source is filtered or to generate a certain single color for illumination. and the camera collects the fluorescence coming out from these cells. And the fluorescence is made by scanning a laser excitation spot point by point on the full surface, and at the same time recording the fluorescence signal coming back from the surface. And this scanning of the surface by an excitation light source is made possible by this instrument, which is called differential spinning disk. So this disk allows the focal spot to scan the surface in a circular fashion to generate a two-dimensional impression of the fluorescence frame. And what does a fluorescent image look like? Here are examples of fluorescence images. What you see in green here in the dark black background are fluorescently marked cells. The cells do fluoresce under excitation, laser excitation. And the camera captures this fluorescence so that we can now optically see the location of the cells. And you see on microcolumns, the cells are relatively uniformly distributed all over. They grow, they say they stick to the cell surface in a very good fashion. They proliferate, they are quite numerous. And this is, again, a fluorescence image. Now, this time we have alternating microcolumns in ellipse patterns. Here in the general view, you also see there is quite a lot of number of cells, but not maybe as many as in here. And if you take a look at this one, in which we have microcolumns within flat surface, you again see quite some large number of cells after 3 hours of incubation. Okay, but this is a relatively early stage, so to speak, only after 3 hours. Now you can see, for example, certain regions in a more careful fashion. You can inspect these regions in a more careful fashion. Here we see a region in which microcolumns and flat regions alternate, and you clearly observe that cells prefer to attach themselves in the early stages to microcolumns rather than flat regions. Of course, there are also some regions in which the cells adhere to flat region, but in this early stage, typically the cells prefer to attach more to the rough micro scale roughened regions rather than the flat regions. Okay, this is the let's say summary of the outcome. And now let's take a look at the live images. What do cells do on, to start with, we take a look at flat polished surface. This is flat polished silicon surface. You see some of the cells move around, they explore around. Some of them move much more rapidly around, and some of them are not as quick. These may correspond to different stages of the cell life cycle. And some of them have grown their axons. They are much more mature than the rest. And some of them are still exploring around to see, to decide where to sit if they want to grow their cones, etc. So we see many different stages of the cells in one frame here. But all in all, the cells are happy, they are live, they are active, they are moving around. Okay. Now, when we take a look at the in case in which we have trench lines, microcolumns within the flat, you see we mark the direction of the trenches. Some of the cells tend to stay in the trench, some of them tend to stay in between the trench, but cells generally prefer to stay in the region where there are trenches and not as much as in the flat. Now, this is a snapshot just showing the alignment of the cells, and you can use this image to see how many of them are in a trench, how many of them are in between, for example, in order to make statistics. This is the same thing, but this time the surface is composed of microcolumn lines with in between ellipse surfaces. And you can see certain regions in which the cells move in a directed fashion quite rapidly, much more rapidly than the rest. And we think that these microcolumns do direct and guide the cells, and they can move more rapidly towards these within these trenches. Now, if you also plot the alignment of the trenches and the gaps in between, you see that most of the cells are aligned within the trench, but there are also a few which are in between, but the statistics tell that the cells would like to adhere more to the, or let's say they are more mobile, they are more exploratory. You see here travel distance on the flat. They are then less exploratory in ellipse and even less exploratory in microcolumns. So in microcolumns, they like where they stay. They don't want to move around too much. Okay, this is moral of the story. And it reflects itself also in the velocity. And they are quite fast in their exploration on flat surfaces, not as much on ellipse surfaces, and even less on the microcolumn surfaces. So their motion is definitely modified by the topography. Okay. Now let's take a look at the late stage cells. Those were the early stage 3Rs. These are the late stage cells results. You see here in the later stages, several cells have been grown into fully, let's say, adult neuron, maybe not fully, but they're much more mature than the rest. And when they are more mature, you see that their extremities align very well to our structuring. So they like to align to deep laser trenches. They prefer to grow their extremities in these trenches. Now, if you do the same experiment, this time on lips versus microcolumns, you observe a similar behavior. Their matured and grown extremities tend to align within the microcolumns, as we have observed before. Most important difference between this and the 3-hour result is that we see a lot of clusters of cells. So after so many hours, the cells start to make they proliferate and they make a kind of colonies. And they are much more mature, some of them at least, than the rest. Now, and if you compare it to a highly structured substrate in which you scratch the surface, scrape the surface with the laser very much, where there are a lot of debris and inorganized kind of topography, then the cells are not as healthy. You don't see many cells which have come to adulthood. Let's say they have not shown growth of their extremities, which is a sign of being healthy and happy and growing into more neuronal direction. So we don't see that in here. So such surfaces are less favorable for the cells, to sum up. Now, if you take a look at the border in between a flat and microstructured, now micro and nanostructured surface, you see now in the first 3 hours case, the flat regions were completely empty and cells tend to stick to the microstructures. But in the later stages, we see cells adhering also to flat polished sides as much as the structured side. But here is a big difference. You see the alignment of the cells on the textured surface with respect to the flat. In flat, they have no organization at all. They are randomly located, but in the patterned side, they are very well organized. They align along the columns. It's much more evident in the later stages of the cell growth. Here are also more images of the microcolumn and lips topographies together. You see how well, they tend to align themselves within these microcolumns in between lips. So this sort of surface texturing can be used to direct the cell organization even better than the flat versus microcolumn. So lips versus microcolumn is more advantageous than that of flat versus microcolumn. Okay, so to put these in numbers, we have statistics. You see the percentage of aligned cells on the lines and between the lines. There are definitely more cells on the lines rather than in between the lines. But on flat, the statistics is more or less the same. So flat versus column doesn't direct the cells as much. Lips versus column does direct the cells quite a lot. Here is the comparison for you, 64% versus 47%, which is roughly 50%, okay, let's say. So some side observations, it seems that these cells generate a lot of stress fibers when they're grown on flat substrates. They need to grow these fibers in order to attach themselves well to the surface, but they don't have to do that on structured surfaces. So therefore, there is a definite difference between the behavior of the cell in their growth on these two different kinds of surfaces. Okay, so I come to my conclusions. So in early stages, cells adhere more readily to structured surfaces than polished silicon. And the cell motility is different on these topographies. Cells move with their lamellipodia. I tend to forget these terms. Their let's say hand or feet on the surfaces, on polished surfaces, and they move on polished silicon twice as fast with respect to lips or microcolumns. In the later stages, we see that they adhere to all topographies, but their nature of adhesion is different. Stress fiber formation is observed more on polished substrates. Their neurites, their extremities are aligned more into line topographies, and they do prefer more microcolumn lines. About 64% of the cells prefer microcolumns than the lips region. So these are the conclusions. And they are not as healthy on highly structured laser scribed substrates. And this work that I have been telling you was the master's study of Sarah. She's with us now. And the laser, fiber laser and scan system was maintained by Dr. Pavlo in our department. And the cell studies were made in the lab of Devrim Son in biological sciences department. So, and we also thank Fırat for his help in various different stages of this work. Okay, thank you so much.
Okay, you can unmute yourself now.
Mm-hmm.
Yes, well, I don't have questions about this part.
You can ask your questions to yourself and answer.
But yeah, I'm happy to see, and hopefully it has continued. That would be what I would like to ask, if there is work continuing in the same
direction
project. Yes, in the same direction, or interest in the application.
Fighting against the odds to continue this work. Yes, but there are several students who are being involved in this line of research. But if you're asking if we were able to do what we wanted to do, we haven't yet. But we are working on it. Brad can tell you more why.
I guess there is very nice
many laser-related problems, but they are about to be resolved soon.
Nice. I mean, I believe there is a lot of potential in the applications of photonics in biology, and I still see a lot of research currently on the same topic. There was a question from Tuche just before, and yeah, I remember that I even saw a paper where they had tried to grow two different cells on structured surfaces, and they were able to see that they would localize to different parts of the structures. So it is very possible to try different cell types in structured surfaces, like where different structures are on the same surface, and if they localize to different parts, then that means you can just see the effects of different chemicals or different treatments on the same, like simultaneously. But of course, there's just a lot of trial because there is still no way that we can really predict how cells behave or how any kind of tissue behaves on different things. So a lot of trial, but when something works, that's really nice because the potential for biological applications is really good. And yeah, I guess with laser or other types of structure, we have a lot of a good way of doing a lot of different structures. So there's just a lot to be tried.
Are there any interest in minds in this direction?
Not with cells. Not that I haven't met any group yet who does similar work here. But yeah, let's see.
Just asking. But we should speak, Sarah, sometime.
Yes, Sila.
Okay. So there is one question. In conclusion, if I'm not mistaken, it was implicated that cells on highly structures were not healthy. What was the reason behind of it? Okay. Well, they did. I think they did not adhere to the surface as a healthy cell would adhere to the surface. And when the cells don't adhere in their natural tendency, they don't decide to take a next step and to mature in the right direction. So they are always exploring around. They are not stable. They are still looking for around for extensive time. So once they find that they are happy, they adhere well, and then they take the next step to maturation. This might have been the reason behind it. So it's like if you're, how to say, if you seed a plant in a place, in a pot, and leave it for a long time, allow it to grow, yes, it likes it. But if you move it too frequently from one place to the other, it never takes the time to adapt itself to the environment and go into how to say further stages of growth. Right? It's similar to that in an analogy, as far as I can speculate.
So hocam, you mean by healthy as the exposed stress levels, right? Exposed health, exposed stress levels. You said like when we force to move the cells, they do not like they do not like it, and they are unhappy about it. So they are unhealthy about it. So we define health as like the exposure to the stress levels.
I mean, they don't start certain biochemical reactions which would progress into maturation. And this, I mean, there is always a chemical background in a living cell, right? It's a living organism. It's a biochemical soup, if you like. So certain cues from the environment are transferred into cell to modulate their chemistry. And if a cell is not happy with its cell, with sorry, cell membrane. If the cell is not happy with its membrane, if the membrane is not stable, let's say, but because the membrane is its chemical interface between the cytoplasm and its environment, right? If that interface is not stable enough, then it doesn't start certain biochemical reactions which would lead to further maturation. So I think this is the biochemical reason. I mean, rather than it doesn't have a brain itself. It doesn't think. It works completely based on biochemistry, right? A cell. So there must be some chemical background to this sort of behavior. And as far as I can understand, cell membrane might be the key for initiation of certain biochemical chain of reactions, but it has to start only after the membrane is stable enough. And I think that might be the reason.
Hocam, thank you.
Mm-hmm. I'm not a biologist again, and this is my kind of speculation. But, well, I don't know, Sarah can tell more maybe if I'm completely off, or do I have right to a certain extent?
No, no, you are not off. I would just like to ask that there are just many ways to see if the cell is unhappy and to answer the question. So we did not measure a stress level of the cell, even though there are some indications of stress in cell which are measurable. But in our case, we just looked at the membrane integrity. And if you look at the image presented, you can see that the cells have these tiny blob-like things. So they're not circular, or they don't have a like oval or any kind of similar structure, but they just have some little blobs, which tells you that the membrane or the nuclear membrane is not stable. So that might be the case. But the thing is that to answer this question, you actually have to do the test. You have to say that, okay, this compound is more in the cell, that's why this is not good. But in our case, it was more about finding a kind of material that the cell likes, rather than finding why the cell doesn't like a certain kind of topography.
Mm-hmm.
Okay, thank you.
If that answers.
Any more questions?
Yeah, if there is no any more questions, we can. No, there's a question.
Uh-huh. Well, what other kinds of surface structures? Well, you can think of everything. I mean, we let's say if you're asking those available at hand in the lab, for example, we are able to fabricate black silicon, which is processed by reactive ion etching or metal-assisted etching, rather than laser processing, which have much higher aspect structures, which are composed of much higher aspect surface topographies. They are needle-like, let's say, pyramidal structures. Not exactly pyramid. They are more like, fairy chimneys in Cappadocia.
Okay.
So it's like these fairy chimneys. They're very, how to say, spiky. So I'm not so sure if the cells would love that. It may, well, but we can further process these topographies to smooth out very needle-like structures a little bit, so that they can still be considered for cell growth. It never occurred to me. Thanks for asking, Sarah. I mean, we have also some other structures available at hand on silicon, such as laser-assisted photochemically processed surfaces. So not directly laser processed, but It's a chemical etchant in which we place silicon that gets activated upon exposure by light. It doesn't have to be a strong laser even. And using that, for example, we are able to generate a silicon surface with a lot of pits, like holes, but random holes, random pits. Otherwise, there's a plateau on top. So it's flat mostly, but it has a lot of pits. So that sort of surface could be quite interesting for study of cells.
Yeah,
it just occurred to me. Thanks for asking.
Yeah, I always wondered about this too. And also, if you have the possibility to make like mold polymeric or other material structures.
These are top-down approaches. Well, in my research line, I don't like top-down techniques so much. I like more bottom-up techniques, self-organized bottom-up techniques, which are then feasible for large area fabrication. So I don't like miniature, small area kind of texturing techniques. But these nano imprint lithography techniques are mature enough, which allows to even fabricate, structure a full wafer, full silicon wafer at once. But it has a lot of, it needs a lot of investments, and I don't have the means to invest on that. So I tend to go into more self-organized surface structuring techniques using light. Well, I allow the nature to organize itself more than top-down organization of the structure by brute force. Let's put it that way. Okay. Well, there are several reasons behind that. Cost is one, but I like it that way. My personal, let's say, matter of taste.
Also, maybe intuition. Because in the body also many structures are like self-organized, and one major motivation for studying cells on the structured surfaces to somehow mimic the body and give the cell something they're familiar with. So in which case they would be happier. So that could be very nice to try.
Yeah, I totally agree. Actually, my exposure to biology, let me think about it. When I think about it, my exposure to biology might have led me to think more in that direction than trying to do everything top down. Yeah, but thanks for this suggestion. I will definitely pursue it.
Okay. Thanks, Professor Bek. We will have a 5 minutes break. If you would like to bring a coffee or something, we will start the next session in 1:50, right? Okay, I have mine too. If you'd like to discuss anything, you are allowed to unmute yourself immediately. I will remove the thing that you can open your camera, discuss it with the professor. We can now stop recording, and we have 5 minutes